Welcome to the deep dive. This is where we take some pretty dense source material, like a textbook chapter, and really pull out the core knowledge you need right now. Today, our mission is pretty fundamental. We're looking at how life ensures genetic continuity, basically how every new cell gets the right instructions. Our sources lay out the two big processes for this, mitosis and meiosis.
Right, and they do very different jobs. Mitosis is essentially cloning at the cellular level.
Yeah.
You start with a full set of chromosomes that's diploid, or two-hand, and you end up with two cells that are genetically identical, both two-hand. Think growth, repair.
Okay, exact copies.
Exactly. But meiosis is different. It's a reduction process. It has to take that two-end cell and accurately cut the chromosome number exactly in half, down to N, or haploid.
And that halving is non-negotiable for sexual reproduction, isn't it?
Absolutely essential.
Yeah.
Imagine if it didn't happen. Each generation, the amount of genetic material would just double. Boom, boom, boom. Very quickly, it'd be completely unmanageable.
A genetic traffic jam.
You could say that.
Yeah.
So before we jump into how they divide, let's talk about what's dividing. We're talking DNA, obviously, organized into units called genes. And these genes are packaged onto these structures we call chromosomes. Those are the vehicles carrying the information.
Got it. So let's lay the groundwork. Where does all this happen? What's the cell architecture look like?
Well, the first big split is between simple cells and more complex ones. Prokaryotes, like bacteria, have their DNA, usually a circular piece, just sort of floating in a region called a nucleoid. No separate compartment.
Much simpler.
Very. But we're mostly focused on eukaryotic cells like ours. Here, the DNA is housed inside a dedicated structure, the nucleus. Think of it as a control room. Inside the nucleus, the DNA isn't just bare, it's wrapped around proteins. Most of the time, when the cell's just doing its job, not dividing, this DNA-protein mix, called chromatin, is all spread out diffuse.
Like a bowl of spaghetti, someone once told me.
Huh. Yeah, that's not a bad analogy. But when it's time to divide, that spaghetti undergoes this incredible compaction, organizing itself into the distinct, visible chromosomes we see in diagrams. Oh, and also in the nucleus, there's the nucleolus. That's the factory for making ribosomal RNA, essential for building proteins later. Specifically, the NOR region, the nucleolus organizer region.
And source material points out something cool. Not all the DNA is even in the nucleus, right?
That's right. Mitochondria, the powerhouses, and in plant cells, chloroplasts, have their own separate small loops of DNA. They actually replicate and use their own genetic information, kind of like little semi-independent units within the cell.
Fascinating. Okay, back to the main chromosomes. When they condense, they have a specific look defined by the centromere.
Exactly. The centromere is this constricted region. Its position gives the chromosome its characteristic shape during division. You have different types based on where it sits: metacentric, submetacentric, acrocentric, telocentric, basically middle, near end, or right at the end.
And that defines the arms.
Yep. The shorter arm is called the P arm, P for petite, easy to remember, and the longer arm is the Q arm. This structure is really important for how they get pulled apart later.
Okay, structure makes sense. Now, a really key concept for organisms like us, homologous pairs.
Ah, yes. Crucial. In diploid organisms, which is what humans are up to, and is nearly all our cells, the somatic cells, have chromosomes that come in pairs.
Yeah.
One member of each pair came from your mother, the other from your father.
And they're homologous because they carry genes for the same traits at the same locations.
Precisely. Same gene locations or loci. They might have different versions or alleles of those genes, but the overall map is the same. They carry the same genetic potential.
Are there exceptions?
Well, some simpler organisms like yeast are actually haploid, and only one set, but the big exception in diploid organisms like us is the sex-determining pair. In humans, the X and Y chromosomes. They're quite different in size and gene content, but they have enough similar regions that they can pair up and behave like homologs during meiosis. It works.
Okay, foundation set. Let's tackle the first process, mitosis. You said this is about making identical copies.
Exactly. Mitosis is for growth, replacing old cells, healing wounds. The end goal: two daughter nuclei that are genetically identical to the parent nucleus. Same two-end chromosome number.
But the actual division, mitosis itself, is just one part of the cell's life, right?
Oh yeah, it's actually a relatively short part of the whole cell cycle. Most of the time the cell is an interphase. This has stages within it. There's G1, where the cell grows and functions, and crucially makes the decision whether to divide. It's a big commitment point.
And if it commits.
It moves into the S phase, S for synthesis. This is the phase where all the DNA is replicated. Every single chromosome is duplicated. So after S phase, each chromosome isn't a single structure anymore. It's made of two identical sister chromatids stuck together.
So double the DNA content temporarily?
Temporarily, yes. Then there's a final prep stage, G2, before the main event. And some cells can actually exit the cycle from G1 and go into a resting state, G0. They just hang out, not preparing to divide.
Okay, so DNA is doubled in S phase. Now we hit karyokinesis, the division of the nucleus itself. How does the cell neatly sort out all these duplicated chromosomes?
It happens in stages. First up is prophase. This is where that diffuse chromatin really starts to coil up incredibly tightly. It condenses into those visible X-shaped chromosomes, each with its two sister chromatids. Also in prophase, the nuclear envelope, the membrane around the nucleus, starts to break down. The nucleolus disappears, and structures called centrioles move to opposite ends or poles of the cell, at least in animal cells. They help organize the machinery.
What holds the sister chromatids together?
A protein complex called cohesin. Think of it like molecular glue, holding them tightly, especially at the centromere region.
Okay, condensed chromosomes, no nucleus boundary. What's next?
Prometaphase and then metaphase. This is all about alignment. Microtubules protein fibers making up the spindle emerge from the poles. They attach to a specific protein structure on each centromere called the kinetochore.
So like grappling hooks?
Kind of, yeah. These spindle fibers push and pull the chromosomes until they're all perfectly lined up single file along the cell's equator. This imaginary line is called the metaphase plate. It's a really precise arrangement.
Precision seems key here.
Absolutely. And the next stage, anaphase, is perhaps the most dramatic and usually the shortest. This is where the separation happens. It's called disjunction. There's a protein called shugoshin that protects the cohesin at the centromere. In anaphase, shugoshin gets degraded. This allows another enzyme, called separase, to swoop in and cleave that cohesin glue.
Snip.
Exactly. And it has to happen pretty much simultaneously for all chromosomes. The moment that cohesin is cut, the sister chromatids are released from each other.
And they're no longer called sister chromatids then?
Nope. As soon as they separate, each one is considered a full-fledged daughter chromosome. And immediately, the spindle fibers, driven by motor proteins, start pulling these daughter chromosomes towards opposite poles of the cell. The fibers actually shorten.
So one complete set goes one way, the other identical set goes the other way.
Perfect segregation. That's the goal. Failure here is a big problem.
What happens once they reach the poles?
That's telophase. It's basically the reverse of prophase. The chromosomes arrive at the poles, they start to uncoil back into chromatin, and new nuclear envelopes form around the two sets of chromosomes. The nucleoli reappear. You now have two distinct nuclei in one cell.
But the cell itself hasn't split yet.
Not yet. That's the final step, cytokinesis, the division of the cytoplasm. In animal cells, the membrane pinches inwards, forming a cleavage furrow. In plant cells, they build a new cell wall down the middle, called a cell plate.
And the result is two separate, identical Cullen daughter cells.
Correct. And the whole process is tightly regulated. There are cell cycle checkpoints, particularly at the end of G1, G2, and during mitosis itself. These checkpoints monitor for DNA damage or problems with chromosome attachment.
Quality control.
Exactly. Molecular quality control. Key players are enzymes called kinases, which get activated by proteins called cyclins. If something's wrong, say DNA is damaged or chromosomes aren't aligned properly, these checkpoints can halt the cycle until it's fixed, or even trigger cell death if it's unfixable.
And if those checkpoints fail?
That's where you can run into trouble. Uncontrolled cell division, ignoring the normal signals and checkpoints, is a hallmark of cancer cells. So the precision mitosis and its regulation are vital for health.
Okay, so mitosis, identity, perfect copies. But life needs variety, right? Evolution depends on it. That brings us to meiosis. How does it generate that crucial diversity while also having the chromosomes?
Meiosis is quite the elegant solution, really. It hits two birds with one stone. One, it reduces the chromosome number from diploid 2N to haploid N, essential for forming gametes or spores. Two, it generates massive genetic variation.
How does it create variation?
Two main ways: crossing over and random assortment of homologous chromosomes. And most of the unique action happens in the first of the two meiotic divisions, meiosis the first.
Which is called the reductional division?
That's right, because it's where the chromosome number is actually halved. It starts, like mitosis, after an S phase, so chromosomes are duplicated into sister chromatids. But Prophase I is drastically different.
How so?
Instead of each chromosome doing its own thing, the homologous chromosomes, the one from mom and one from dad, for each pair, find each other and physically pair up side by side. This intimate pairing process is called synapsis.
Okay, they're buddying up.
Exactly. The paired structure, containing the two homologous chromosomes, is called a bivalent. But since each chromosome already consists of two sister chromatids from S phase, this bivalent actually contains 4 chromatids in total. So we call it a tetrad.
4 chromatids all closely associated.
Yes. And it's within this tetrad structure that the first major source of variation occurs, crossing over.
This is where they swap pieces.
Precisely. Chromatids from the homologous chromosomes, so one maternal chromatid and one paternal chromatid, not sister chromatids, physically overlap at points called chiasmata. At these points, they break and exchange corresponding segments of DNA.
Wow, so you're literally shuffling the genetic deck within a chromosome pair.
You got it. You end up with mosaic chromosomes that have new combinations of alleles inherited from both parents on the same chromatid. It's a major driver of genetic diversity.
Okay, so prophase I, pairing and crossing over. What happens in metaphase I?
In metaphase I, it's the tetrads, these paired homologous chromosomes that line up at the metaphase plate. And crucially, how they line up is random.
What do you mean by random?
For each pair, it's a 50-50 chance whether the maternal chromosome faces one pole and the paternal faces the other, or vice versa.
Yeah.
And the orientation of one pair has no influence on the orientation of any other pair. This is the random assortment of the second big source of variation.
So many possible combinations of how the parental chromosomes get sorted.
Huge numbers of combinations, especially with many chromosome pairs, like in humans. Okay, then comes anaphase of first. This is the critical reduction step.
What separate?
The homologous chromosomes separate, the tetrads split up. One complete chromosome, still composed of two sister chromatids, now called a dyad, moves to one pole, and the other homologous chromosome, its dyad partner, moves to the opposite pole.
Ah, so the centromeres don't split here.
Correct. That's the key difference from mitotic anaphase. The sister chromatids stay firmly attached at the centromere. Because whole chromosomes, dyads, are separating, not sister chromatids, the number of centromeres going to each pole is halved. That's why meiosis I is the reductional division. We go from 7 to n in terms of chromosome number, although each chromosome is still duplicated.
Okay, so at the end of meiosis II and telophase cytokinesis II, we have 2 cells, each haploid N, but every chromosome is still an X chip, a dyad.
Exactly. That's why we need a second division, meiosis II.
And this one's called the equational division.
Yes, because the chromosome number doesn't change here. It starts as haploid N and ends as haploid N. Meiosis II looks, mechanically, much more like mitosis.
How so?
In Prophase II and Metaphase II, the dyads in each of the 2 cells from meiosis I line up individually at the metaphase plate. Then in anaphase II, the moment arrives, the centromeres finally split.
Just like in mitosis anaphase.
Just like it. The cohesin holding the sister chromatids together breaks down. Shugoshin protection is gone. And the sister chromatids separate, moving to opposite poles. As soon as they separate, they're called monads, or just chromosomes again.
So N dyads become N monads moving to each pole.
Precisely. The chromosome number remains N. Then telophase II and cytokinesis happen, resulting in a total of 4 haploid N cells from the original single diploid cell.
And each of those 4 cells is genetically unique because of crossing over and random assortment.
Absolutely unique, ready to become gametes like sperm or eggs or spores, depending on the organism.
Speaking of sperm and eggs, gametogenesis, the source material highlights some really striking differences in how meiosis plays out in males versus females, doesn't it?
Oh, hugely different outcomes. Even though the chromosomal events are the same, it all comes down to how the cytoplasm gets divided.
Okay, tell us about spermatogenesis.
In males, in the testes, a starting cell called a spermatogonium undergoes meiosis. Both meiosis I and meiosis II involve equal divisions of the cytoplasm. The result is 4 cells of roughly equal size, which all develop into functional motile spermatozoa, or sperm. Very efficient production.
4 functional sperm from 1 starting cell.
Yep. Now compare that to oogenesis in females in the ovaries. It starts with an oogonium. Here, the meiotic divisions are profoundly unequal.
How unequal?
In meiosis I, when the cytoplasm divides, almost all of it goes to one daughter cell. The other cell gets a set of chromosomes, the dyad, but virtually no cytoplasm. It's called the first polar body and usually just degenerates.
So 1 big cell, 1 tiny one?
Right. Then the big cell proceeds to meiosis II. Again, the division is unequal. Most of the cytoplasm goes into the one cell destined to become the ovum, the egg. The other tiny cell, the second polar body, also gets discarded.
So from 1 starting cell, you only get 1 functional egg.
One single, very large, functional ovum. The inequality is purposeful. It ensures that the egg cell is packed full of nutrients, organelles, mitochondria, everything the developing embryo will need in its very early stages before implantation. The polar bodies are just a way to get rid of the extra sets of chromosomes while conserving resources for the winter.
That makes sense. And in humans, oogenesis can be paused for a very long time, right?
Yes, it's quite remarkable. Meiosis at first starts before birth, pauses in Prophase I, resumes at puberty, pauses again in Metaphase II, and only completes if fertilization occurs. It can be arrested for decades.
Incredible complexity. Okay, one last piece from the source material. We keep talking about chromosomes condensing, going from that invisible chromatin spaghetti to visible structures. What's actually happening physically? How do they pack so tightly?
It's an amazing feat of biological engineering. That long, thin chromatin fiber has to be condensed enormously to become a short, thick, mitotic chromosome. The source mentions something like a 5,000-fold compaction in length.
5,000 times shorter. How?
The model described is the folded fiber model. It suggests that the basic chromatin fiber itself undergoes extensive coiling and then folding, looping back on itself in a highly organized, non-random way. Think of coiling a rope, then coiling the coils, and then folding those coiled coils.
So it's a very structured packing process.
Extremely structured, it has to be, to ensure that the chromosomes have that consistent shape we see during mitosis, and crucially, so they can be pulled apart during anaphase without getting tangled or broken. That orderly structure is key to function.
So we've journeyed from the cell level down to the molecular packing of DNA. Bringing it all together, what's the big takeaway for us, the organisms built by these processes?
I think the takeaway is the duality. You have mitosis, this engine of unbelievable fidelity, ensuring that when your skin heals, the new cells are identical copies, maintaining tissue integrity, cellular identity. And then you have meiosis, which embraces risk and change. It has the genetic content precisely, but also shuffles the deck through crossing over and random assortment, generating the variation that allows species like ours to adapt and evolve over generations. Species continuity and diversity.
Fidelity and variation working together. Okay, let's end with a thought for you, our listener, to ponder. Given the incredible molecular machinery we've discussed, the checkpoints, the motor proteins, the enzymes like separates, what do you think are the immediate downstream consequences of just one tiny error? Say one pair of homologous chromosomes failing to separate an anaphase of first, or one pair of sister chromatids failing to separate an anaphase second. That event is called non-disjunction.
Yeah, that single failure. It seems small, but it means the resulting gametes will have the wrong number of chromosomes, either one too many or one too few. And the source notes that things like radiation or certain chemicals can increase the chances of these non-disjunction events. When such a gamete is involved in fertilization, the resulting embryo has an abnormal chromosome number, which often leads to significant developmental issues or genetic disorders. A tiny molecular slip-up with potentially massive organism-level consequences.
Something to definitely think about. Thank you for joining us on this deep dive into how our cells manage their precious genetic cargo through mitosis and meiosis. We hope the difference between the copy and the shuffle is much clearer now.